All right, well, welcome to Lecture 8. This is our second of 4 lectures on development, and this is the second part of my lectures on adolescence. What I'm going to do today is focus on 3 specific areas of adolescent development that has to do with social neuroscience, and that will be emotional processing, risk-taking, social acceptance, and exclusion. Now, all 3 of these topics, I'm going to really drill in on specific research studies. And this gives me a chance then to kind of show you how a specific study might be done on topics like this. And so I'm going to tell you a little bit more about the methodology that I've done in the past, and I'll also talk about some of the limitations of each of the different studies. And then finally, I'll end up talking about sort of like a reconsideration of adolescence, like what should we do as a society or as parents or teachers to help out adolescents. All right, let's take that first topic, emotional processing, okay? Now, emotional processing or emotions just generally in adolescence has been something that adolescent researchers have been interested in ever since it became sort of a research topic in psychology. And we can trace a lot of this back to Stanley Hall, this guy you see here in the picture, who was a very prominent American psychologist early in the history of psychology in the United States. He was a president of the American Psychological Association. He started one of the first graduate programs in psychology. So he trained many, many people, and he was a leading researcher on development. And you can see that in 1904, he wrote a book, was published, and it was called Adolescence, but its full title was actually Adolescence, its Psychology and its Relations to Physiology, Anthropology, Sociology, Sex, Crime, Religion, and Education. And so in some ways, it's almost like a title you'd expect from a social neuroscientist in the sense that he's looking at different levels of analysis. Now, in this huge opus of a book, he does give definitions of adolescence. He talks about what we knew about at the time in terms of research on adolescence. But one of the kind of phrases that always comes out when people talk about Hall's approach to adolescence is this one, that adolescence is a period of great stress and strain, storm and strife. Right? So that suggests that adolescence is just full of like emotions out of control, and things are really very difficult emotionally for adolescents. Now, we're going to find that this isn't really the case. I mean, sure, there's stuff going on with emotions in adolescence, and there's a developmental phase that adolescents are going through to try to, better handle their emotions and so on. So in order to think more about it in a modern sense, I want to show you this model of emotion regulation and emotion processing put forward by James Gross and Thompson in 2009. Now, James Gross is a psychologist at Stanford University. He's for several decades now done a lot of work on emotion and emotion regulation. And what he does is he kind of breaks emotion down into these different phases that you can see here. So we have the situation, and then you have attention and appraisal, and then the response. And under emotional regulation, which is the big box on the top there, he's trying to say if you're trying to regulate your emotion, like if you're trying to have sort of maybe you don't want to laugh during a serious moment, or you're trying to not cry when you want people to think you're not upset, you can do a couple different things to regulate your emotion. One thing you could do is you could affect your concentration, your attention. And so you can see here that we have attentional deployment concentration, where you kind of really focus on what the emotion is, or you could try to distract yourself from paying attention to an emotion by focusing on something else. Like in the sense of an experiment, you could say, instead of focusing on the emotion of that face, focus on the gender of the face. But either way, the idea is that you are regulating your emotion through the way you attend to the picture or attend to features about the emotion or the emotion stimulus. And then the second step that you can see there is what's called appraisal. And this is how you're going to basically handle the stuff that you just got through the attention phase. So you could have a way for you to tell yourself that you're going to not have a big reaction to this. You could say to yourself, hey, that's just a fake photo. The person isn't really having this. Or I'm watching a movie and I tell myself this scary movie isn't really happening the way it looks like on the screen. Those are just actors, whatever. So appraisal is when you have this kind of deeper level of thinking, and that could also help you with your emotion regulation. And finally, you have your response. Emotional reactivity that is down the bottom is just sort of more of your natural response to having that stimulus presented to you. And you can just see there, you view a photo and you have a reaction to it. Now, what I'm going to be doing is talking a little bit about emotional reactivity, but I'm also going to be focusing on this part about how we identify emotions. So we're kind of focusing today just on these early stages of how people or adolescents respond to emotional stimuli. But you could look at differences in adolescents and versus adults on how they appraise things, how they manage their emotions. There's just much more we could do, but I just wanted to just give you a little flavor of this research on emotion by focusing on the differences between adolescents and adults. So I'm going to show you this study here by Vink et al. That was conducted in 2014, title which was Functional Differences in Emotion Processing During Adolescence and Early Adulthood. And what they did, this study was done in the Netherlands, and you can see they had 60 Dutch participants who were between 10 and 24 years old. They had 28 females, 32 males. And while they were in an fMRI scanner, they viewed positive, negative, and neutral pictures from the International Affective Picture System. It's also abbreviated as the IAPS. These are examples from the IAPS. So you can see these are pictures like the one in the upper right corner. I would assume most people are going to find that to be a negative picture. Maybe the baby picture and the picture of the animal there is going to be a positive picture. And maybe that picture of that guy just looking at the camera there is sort of a neutral picture. All right, so there's a huge database of these pictures in the International Affective Picture System. They're just still photos that have been rated by hundreds of other participants, so we kind of know what the typical rating is for that picture in the upper right, what the kind of picture is rating is for a picture of a baby, and so on, in terms of how positive or negative or neutral the picture is. So in the Vink et al. Study, they used those stimuli, presenting those pictures one at a time while the person's in the scanner. And then after the picture was shown, they indicated, the participant, whether it was neutral, negative, or positive. So they didn't have to rate it. They just had to say with a button press, was that a neutral picture, negative picture, or a positive picture? All right? So they're going to go ahead and scan people's brains from 10 years old up to 24 years old while they look at different emotional stimuli. And so now if you could imagine that you're in the researcher's shoes and you're about to analyze your data, or actually you probably want to do this before you even ran the study, I want you to think a little bit about which areas would you expect should be activated during this kind of task when you're looking at emotional stimuli. That is, what parts of the brain, from what we've already covered in previous lectures, would you expect to be involved in emotional processing? So in fact, what they were interested in was the amygdala. So they looked at amygdala activity. They looked at the hippocampus. They looked at the ventral striatum, the orbital frontal cortex, the OFC, and the ventral lateral prefrontal cortex. All right, so these are the main areas: the amygdala, hippocampus, ventral striatum, orbital frontal cortex, and the ventral lateral prefrontal cortex, because all of these areas of the brain have been found in previous research to be involved in emotional processing. So you can see then the main hypothesis is that they're going to look across development, look from the participants who are younger to the ones that are older. And what they expected was that frontal activation would increase as you got older. So you would use more of your prefrontal cortex when you were processing these stimuli, and your limbic subcortical activation, like your amygdala, that activation would decrease, because they argued that you have sort of like this more emotional, immature response when you're younger, at the beginning of adolescence, where it's primarily your limbic system saying, fearful or happy or whatever. And then you don't have much prefrontal activity yet. You don't have a good prefrontal cortex, so you can't really manage it. So it's really being determined more about your limbic system. But as you grow older, you can tell yourself, I'm looking at this picture here. That's just a picture of some bugs. And you have more frontal activation, and you kind of appraise it a little bit differently. You can distract yourself a little bit differently and not necessarily have this sort of rampant limbic activation going on while you look at these different kinds of stimuli. All right, so that's really taking this sort of complicated study and just boiling it down to its simplest elements. So what did they actually find? Well, they first of all found that their participants could correctly identify the picture as positive, neutral, or negative. That is, they scored about 80% or higher on those pictures. So when a positive picture was shown to them, about 80% of the time they indicated it was a positive picture, neutral picture, and negative picture. Interestingly, they didn't find any difference between the ages on this. So all ages did this equally well. So there was no developmental changes in how the participant categorized the picture. Now, across the main regions of interest, or they're also called ROIs, these main areas that I was telling you about, there was no difference when you compared how people responded to positive and negative pictures. So the areas that I'm going to tell you about don't differ between positive and negative pictures, but they differ when you contrast them between like a positive picture and a neutral picture, or a negative picture and a neutral picture. Now you remember that this is that method of subtraction. So in fMRI, you always need to have something that you're comparing your response to a stimulus to. And so in this case, what they do is they analyze what kind of brain activity is involved with positive pictures compared to neutral, and what brain areas are involved with negative pictures and neutral, okay? Because they didn't find any difference when you contrasted positive and negative to each other. All right, so here's what they found. These are the three biggest areas that they really focused on. And you can see here that we have left on the top and right on the bottom. So this is left and right hemisphere. We have the amygdala, we have the hippocampus. So those are our more limbic structures. And then we have our ventral lateral prefrontal cortex over on the right, okay? And you can see that the red and the black indicate what things are significant and what things are not significant. You can also see, negative is actually the black dots, positive is the red dots, and then they can also tell you whether or not some things are significant, okay? So what they found here is for amygdala and hippocampus, positive and negative pictures had highest activation compared to neutral in younger participants. So you'll notice the slopes there in the amygdala and the hippocampus. They're going down for positive and negative pictures when you compare them to neutral. So that is the younger participants, the ones that are closer to 10, 11, 12, 13 years of age, they're the ones that have bigger activation there in their limbic system, right? And then as they get older participants have less activation in the amygdala and the hippocampus for both positive and negative pictures. The p-values, by the way, are a little different here and there. Some things are below 0.05, some things are not below 0.05. But overall, I want you to just kind of get a sense of the trends of the data here for amygdala and hippocampus. Now, what about that frontal area, the ventral lateral prefrontal cortex? Well, what they found there was that they get more activation for positive pictures in older participants. So they didn't really get much of a difference there for negative, and it was only really in the left, but they are seeing that the older the participant, the more likely it was that they were able to show activation in the ventrolateral prefrontal cortex when they looked at these positive pictures. So it sort of supports their hypotheses, right? That the younger participants in the study are doing more limbic activity, the frontal cortex, the ventrolateral prefrontal cortex, particularly on the left, is more activated in these older participants, specifically for positive stimuli. Now, another thing they did in this study that they analyzed was they looked at what's called functional coupling, where you can look at co-activation between different areas. And what this means that what you try to show is that across different kinds of stimuli, do you see different areas seem to be connected to each other, that they light up at the same time, they become more active. And so generally, here's what they found. With older participants, you found more coupling between the amygdala and the medial OFC. So younger participants don't really show this coupling, that when the amygdala goes off, the medial OFC does, but older participants do. Which suggests again that, sure, maybe the amygdala is having a response, but the medial OFC, this prefrontal area, is showing maybe some sort of dampening down of the emotional response. There was more coupling between the right hippocampus and the left medial OFC. So again, another connection between like a limbic structure and in the frontal cortex. And there was more coupling between the right hippocampus and the left ventrolateral PFC. So again, don't focus too much on whether it's left or right and whether it's the OFC or whatever. I'm just trying to show you that as you get older, these participants seem to be showing more of a connection between that limbic system structure and the stuff going on in the frontal cortex. Now, that's the study in 2014, but I want to point out a few sort of caveats or things to consider when you think about the results of this study. No study is going to have all of the answers. No study is perfect. But let me point out some sort of weaknesses and other interpretations of the study. First of all, keep in mind that for their emotional stimuli, they're only using IAPS pictures. These, sure, they're pictures that have been rated by hundreds of other people, but they are still photos, right? So they're just pictures of still images that are presented to the participant. They're not particularly emotionally strong of any sort. It's not like seeing pictures of, your mother or somebody you love very much. It's just still photos of things that come from a big database. And they're still. They're not actually videos. They're not sort of interactive with words and things like that. Another thing that's really important to notice is that even though we're talking about some sort of changes between adolescence and adulthood in the way the brain responds to the stimuli, it made no difference in the actual behavior for the participants. So remember I told you that when they made choices about whether this was a neutral, a positive, or a negative image, there was no difference across the ages. They all pretty much got it right. So that kind of suggests that perhaps they didn't have the actual best behavior, a best rating that could be used to help us understand how these brain regions might be involved in the way we process emotional stimuli. Another thing is they didn't actually assess any puberty changes. So for those really young participants, some of them would have been going through puberty and hormones would have been coming into play and would really possibly affect their emotional responses. But in the Vink et al. Study, they didn't even measure this. They didn't pay attention to it as part of development. And the other thing to keep in mind, it is a cross-sectional study. So everybody's being tested at the same time at all these different ages. It would be a stronger kind of study to do this as a longitudinal one, where you could maybe take 60 10-year-olds and follow them up every year and watch as they get older and watch how they change, rather than trying to say it's something across these different ages in the way that this study was done. Nevertheless, I think it's still interesting, and it starts to give you some ideas about how social neuroscientists who use fMRI could look at how emotional responses occur. I'm going to give you one further wrinkle, one more thing to consider about this kind of research. Now, I didn't really differentiate in those stimuli from the IAPS, whether the stimuli were social or not, but you noticed that some of those stimuli had people or babies in them. Other stimuli just had like a cockroach or an animal. Does that matter? Does it matter if the emotional stimuli are social or not? In other research, it does really make a difference. You can actually find studies where they've used the social pictures from the IAPS versus non-social pictures from the IAPS to look at brain differences. Here's the study, though, conducted by Burnett et al., published in 2009 in the Journal of Cognitive Neuroscience, where they tested 19 adolescents who were between 10 and 18, and then 10 adults between 22 and 32, while they thought about scenarios involving social or basic emotions. So they didn't use pictures. They don't have IAPS. They just have things that you sort of imagine yourself being in with situations. So, for example, some of the social stimuli, some of them involved embarrassment, some of them involved guilt. But, like, for instance, you're supposed to imagine your dad started doing rock and roll dances in the supermarket, or you tripped over in front of a boy you liked, or you were eating with your friend and you dribbled down your top, or you laughed at a quiet girl and it made her sad. So those are all emotional stimuli that are social. And then the other kind of stimuli they have here are more the emotional responses about something more basic that isn't social. So you saw your friend was vomiting next to you and you could smell it. So it's about the vomit itself that you're having the response to. Or you saw a big hairy fly laying eggs on your friend's lunch. Or your dad told you that the fridge was infested with maggots. Now, you'll notice that they do mention your dad, your friend, and so on. It sounds like it's social, but the emotional response of like disgust or fear is about something else in the actual scenario. And the only reason why they mentioned their friend and dad was just to have it sort of be like a control, that they had the same sorts of words appearing in their basic emotional stimuli as they did with the social stimuli. All right, so what did they find? Well, they found that when they looked at the differences between social stimuli versus basic emotional stimuli, they found differences like in the left temporal pole. Okay, so back near the TPJ, basically, in the temporal lobe, you can see that as the participant gets older and you're trying to look at a difference between social stimuli and more basic emotional stimuli, you're getting more activation in the temporal lobe, or temporal pole, in the left temporal pole. Over there on the right, you can see the medial prefrontal cortex, right? And so there, what's interesting is that when they're younger, they're showing more MPFC to social stimuli, and as they get older, they do this less so. So what that suggests is that, well, first of all, adolescents activated a lateral part of the MPFC for social versus basic emotions, whereas adults did not. And relative to adolescence, adults showed higher activity in that left temporal pole for social versus basic emotions. And so here's what they conclude. They say, well, although the MPFC is activated during social emotion in both adults and adolescents, adolescents recruit anterior MPFC regions more than do the adults do. And the adults recruit posterior or temporal regions more than the adolescents do. And so what they're saying, as you develop, sure, one of the first things that you're going to be using a lot to help you make sense of social emotional stimuli versus basic emotional stimuli is your MPFC. This is the part of the prefrontal cortex that's going to be involved in a lot of social understandings of relationships. But as you get older, as your brain matures, more of that work about making sense of social emotions and social situations gets pushed back more towards the temporal region, to these posterior regions, where we process things like theory of mind and face processing and so on. So they're trying to make an argument here that development across adolescence is sort of like changing the focus of processing from the medial prefrontal cortex for social emotional stimuli to more posterior regions. And that's one of the things that happens during development. And I think that could be partly due to, like I said, whether or not you're really good at perspective taking. So perhaps in the early years, the MPFC isn't really, it's helping us make sense of social situations, but it's not really about perspective taking and trying to understand why this person or why this thing in this social situation would be this way. And that would require more of these posterior regions that they think are involved in the social brain for adults. All right, so that's our first topic of interest today, is on emotional processing. Let me move now to risk-taking. Risk-taking is also talked about in Sapolsky's book. Now, the main thing here to think about is that the adolescent brain is really hypersensitive to rewards. And risk-taking then is really about that hypersensitivity to rewards. It's more prevalent in adolescents and young adults than it is for any other age group. And I'll point out here, and it's only worse for people who are sleep deprived. So you're even more likely to take risk when you're sleep deprived. So if you compound that with adolescents and young adults who are sleep deprived, then we're really talking about a lot of risk taking going on. What are examples of this kind of risk taking? Like dangerous driving, like texting while they drive, drug use, binge drinking, that should have been binge, not bing, and risky sexual behavior. Why? Why would this risk taking be more prevalent in adolescents and young adults than any other age group? Well, perhaps it's because of the ventral striatum. Like if you saw in that last lecture, we talked about how Blakemore had done work on decision-making in adolescents versus adults. It could be that the ventral striatum is just so profoundly pumping out responses here that the prefrontal cortex can't actually manage, that the ventral striatum's need for reward, for positive positive rewards and positive stimulation is what drives a lot of that risk-taking. So we have that still developing PFC, and importantly, the lateral PFC, which seems to be really important in managing, our need for reward. So I'm going to give you another example of this. I talked about this in the last lecture about how Blakemore has used a driving task. Here's another one. This is Van Horn et al. In 2018, differential effects of parent and peer presence on neural correlates of risk-taking in adolescence. So this one involved only adolescents, early adolescents, people between 12 and 15, 56 of them. And they played what's called the yellow light game while they were in the scanner. Okay, and the goal is that you're going to drive a virtual car through all of the intersections in the shortest amount of time. So they're in the scanner, they got like a little button press thing so they can make decisions as they get to different intersections. At each intersection, the driver chooses to go through a yellow light. So there's a yellow light there at the intersection. Or they can, so that would be go through the yellow light, or they can brake and stop and wait for the lights to change again. All right, so it's whether or not you want to blow through those yellow lights or you're going to stop every time you see a yellow light. And about 50% of the time, if you go through that yellow light, another car is going to cross through the intersection and crash into you. And if there is another car and it crashes into the driver, that driver then gets a 5-second penalty before they can resume again. So you're risking, sure, maybe you'll get through that yellow light and speed through and everything will be fine, but you're also risking that you'll get a 5-second penalty that will slow down your overall game if you choose to go through a yellow light. And so in this study, these adolescents played the game with either a peer present, so they had another friend of theirs there, or their parent present who was watching them in the scanner. right? So they're not in a scanner themselves, but they're nearby. And so it's either a peer present or a parent that's present. So they're controlling for the fact that there's someone there by having a peer or a parent present. This is an illustration that comes from the article of that yellow light game. And you can see, so you come up to an intersection there up at the top, and it's got a yellow light. And so now over there on the left, you could decide to go. If you're successful, you can just speed right through. You're going to have no delay. But about half the time, you're going to crash into somebody, and you're going to get a 5-second delay. You could stop. And by the way, if you stop, you're going to have to wait 2.5 seconds before the light changes again. If you can see that a car went through that, you're going to go, oh, that was great. I'm glad I stopped because I would have crashed into them. But again, about half the time, there's no one there. So if you stop, it's sort of like an unnecessary stop. And finally, they also had the option that people sometimes would have a no decision. They could decide not to do anything. And in this case, they suffered a 1-second delay if they didn't actually press the button. They tried to not have that happen as much as possible. They really wanted people to make a go or a stop decision. And then you can see the social context is that you're either doing this with a peer present or you're with your parent. And of course, it's not actually like that in those pictures, but they're in the scanner and their partner is nearby somewhere. All right, so what did they actually find? So we can look here and see two main findings. And so up at the top, we're looking at ventral striatum and the temporal parietal junction, okay? So when participants choose to go through a yellow light, you can see that the ventral striatum and the TPJ were greater in the peer condition. So that's what that's showing us there, is that peer versus a baseline is greater. So that means you're getting more ventral striatum activity when they decide to go through a yellow light. They don't get as much ventral striatum when they go through a yellow light and their parents present. And they also get more TPJ. Remember, this is that kind of mentalizing part of the brain. That's also higher when they decide to go through the yellow light compared to when their parent is present. Now, they could decide to stop at the yellow light. So what about the participants when they stop at a yellow light on those trials? What's going on? And what we can see there is we really get a difference between stopping at a yellow light is more for the DLPFC. So don't pay attention too much to the fact that the parent baseline is negative. What the real key thing here is to show is that we're getting more DLPFC for these adolescents when they decide to stop at the yellow light and their peer is present. So it's kind of like they have their peer present, and it's usually rewarding to just blow through there, like you can see up at the top. But if they do decide to stop, it's because they're getting more DLPFC, more prefrontal activity to kind of shut down that wanting to get rewarded by blowing through the yellow light. And this is particularly strong when they have a peer present. So the peer matters more than the parent does, obviously, in all these three things that you see here in terms of these responses. Now, that's a really interesting study, and I think, again, it shows you a little bit about the risk-taking of adolescents versus adults. A few caveats, though, a few things to think about. First of all, when they looked at the actual behavior of whether they went through the yellow light or not, the behavior didn't show the effects as predicted. They had three conditions, really. They had the alone condition, where you could be by yourself making these decisions. You have a peer condition, where your peer's nearby or your parent's nearby, right? So you would think that based on the data that when the peer was present, they would get the most go decisions, right? They're going to make these really risky decisions. But in fact, they did this the most when they were alone. So what this shows us is that their behavior didn't fit here. They were actually less risky when their peer or their parent was there compared to when they were alone, all right? And no comparison group and age range. There was no comparison group here. So we don't have adults. We only have adolescents. So we don't know if on this particular task if adults would be different. We also don't have much of an age range here. It's a pretty narrow little band of adolescents that we're looking at. So we can't really see how this changes across adolescents in this particular study. Now I'm going to show you one more study that I think is relevant to all of this. And this is a really interesting study that was done at UQ, at the University of Queensland, back in 2010 by a PhD student named Richard Ronay at that time, who's now a professor, I think, at Columbia, and his advisor, Bill Von Hippel, who's still on our staff at UQ. Now, what they did in this study is it was, by the way, the title of it is The Presence of an Attractive Woman Elevates Testosterone and Physical Risk Taking in Young Men. It was published in Social, Psychological, and Personality Science. And what they did is they actually went out to Brisbane skateboarding parks to collect their data. So this is a nice field study out there in skateboard parks. And they had a couple independent variables, things that they were actually asking the participants to do. One is they would ask the participants to perform on their skateboard an easy or a difficult trick. And they would just say to the participant, I'd really like you to pick something next that's an easy trick for you. And I don't care what it is, just something that you know you can do like 100% of the time without making a mistake. Or could you do a difficult trick for me, something that maybe about half the time you actually crash and don't actually succeed in performing the trick. The other independent variable was who the experimenter was that was asking the participants to make these tricks. And so you could either have a male experimenter who did it the whole time, or about halfway through the experiment, the male experimenter would switch and you'd get this attractive female that they used for all of the participants. And apparently she was this very attractive undergraduate at UQ, and they brought her out to a skateboard park at this particular moment where she would then suddenly take over running the experiment. The dependent variables included, they actually looked at that skateboarder's attempts at the easy or difficult trick, and they looked at whether or not it was successful, whether the person crashed, or whether they kind of like chickened out in the middle of it. They aborted their attempt at the particular trick. And they also measured their testosterone. They did this at the end of the study. They also asked them to do what's called a reversal learning task, which was a proxy for ventral medial prefrontal cortex functioning. That is, they couldn't actually put people in a scanner here. So instead, they gave them a little task that basically was done on a laptop where they had to make risky decisions about blowing up a balloon. And if you blew it up too far on the balloon, you would make it pop and you would lose any tokens that you would want up until that point. And so the point is, if you're sort of a risk taker, you're not really showing much control over your risk-taking ability here, you're going to be making the balloon pop more and you're going to end up with fewer tokens. All right. Now, why did they do this study? Interestingly, their study was done to test hypotheses that are derived from evolutionary biology. So they were talking about the fact that they thought that attractive women have the power to shift men's time perspective away from the long-term consequences of their choices and focus their attention on the here and now. So they have good reasons they outline in the beginning of that paper about why evolutionary biology would say this. But basically the idea is that if you're a young male, you're thinking about mating. And if you see this attractive woman, you're not really thinking about the long-term consequences of being injured. Instead, you're maybe focused on right now impressing her and taking bigger risks. And so they have a nice sort of theorizing about this in their paper. We're going to talk more about this sort of evolutionary approach to social behavior in lecture 14. But the other thing to consider about this, besides the way that Ronne and von Hippel think about it, is that the participants were actually young males, okay? And their mean age was 21.58. So they started with 18-year-olds that they found at the park. And I noticed that if you kind of look at the age distribution, it looks like most of them are very young adult males. I wonder if even some under 18 slipped into the study somehow by lying that they're 18. I mean, if you go to a skateboard park, I'm guessing that it's generally going to be adolescents that are skateboarding there. You're not going to find a lot of older 20-year-olds and 30-year-olds in there. But anyway, just keep in mind that the point is here that they're using fairly young males here who maybe have still developing prefrontal cortexes. So this could be considered then a study of late adolescence where the PFC isn't fully developed. In that case, then what we might expect is that the ventral striatum is still ruling more, right? It's still not the case where the MPFC would have its sort of shutdown of ventral striatum motivation. So social rewards, including the attention of attractive females, might be appealing to this still developing young heterosexual male brain. And I've noticed that I am going to say it's a heterosexual male brain because this is all working under the assumption that the reason why that attractive female has her effects is that these young males find her sexually attractive. All right, so if you take that just then as a test of late adolescence. I think that it doesn't say anything is wrong about the way Ronne and von Hippel did their study. It's just saying this is another interpretation of what you might expect to find here. All right, so here's what they actually found. Here's the results. And so what you can see is they've classified the easy and the hard tricks, all right? And they're only looking here at the difficult tricks. So in block 1 and block 2, they have the first 10 tricks and the second 10 tricks. So these are 10 difficult tricks and then second 10 difficult tricks. Some of these guys have a male experimenter for both the first and the second blocks. You can see the little symbols there that up in the upper left, for instance, on aborted tricks, you can see that we get the same number of aborted tricks in the first 10 and the second 10 when there's a male experimenter. But notice that when we get to the second kind of participant you have there, where they were assigned to a condition where a female experimenter is doing the second 10, that you can see that we get fewer aborted tricks. So that is when she is there looking at the second 10 tricks, these guys are performing for their difficult tricks fewer aborted tricks. That is, they go through it, right? And in fact, what you see then over on the right is that what's going on is that when there's a attractive female there, they're actually making more failed tricks, okay? Also, if you look down at the final graph, you can see that they're making fewer successful tricks when they have a male experimenter in the second 10, but they're making more successful tricks when there's a female present. So they're being riskier. That means they're getting more failed tricks, but they're also getting more successful tricks when that female experimenter is present, compared to those participants who were randomly assigned to have the same male experimenter for all 20 tricks. I hope that's clear. I think it's a clever way of looking at behavior, just looking at these skateboarders and asking them to define what they thought was a difficult trick for themselves. Now, remember I told you that they measured reversal learning, they also measured testosterone, and one of the things they found was that there was a relationship here between the testosterone and the experimental manipulation of the gender of the experimenter. So you can see that yes, there's this relationship between the experimenter's gender and aborted tricks, that when she's present, they have fewer aborted tricks, right? So that's why there's a negative relationship there between one and the next. But this relationship was mediated by testosterone. And what we find there is that those participants who had the female watch him for those final 10 tricks had increases in testosterone, which then caused them to have fewer aborted tricks. And so there was a causal pathway there where testosterone mediated the effect between the experimenter's gender and the aborted tricks. So that's kind of clever. It's kind of an interesting way to show that their hypotheses were supported here, that these males are really interested in the short term, their testosterone levels go up, and then as a result of that, that's why they make fewer aborted tricks. They go through with it, make more successes, more mistakes, but they don't chicken out. So how do we interpret this? Well, obviously Ronay and von Hippel thought this supported their hypotheses. But a couple things to notice here. First of all, is that testosterone was only measured once at the end of the experiment. So we don't know if there were pre-existing differences in testosterone before the experimenter began. So that is, it could have just been that maybe the guys who were in high testosterone are the ones where the female experimenter was assigned. I mean, it's unlikely since they used random assignment, but we just don't know because they didn't measure testosterone at the beginning of the study. We also don't know exactly what during the experiment caused their testosterone to go up. Was it the presence of the female? Was it just because they changed experimenters that caused their testosterone levels to go up? We don't know, because all we have is really this sort of blunt measure of testosterone measured at the end. Now, the reversal learning task, I haven't mentioned that yet, but what they did find was that people were better at this task when they did it in front of the male experimenter than in front of the female experimenter. That is, they didn't blow up as many balloons and they ended up with more tokens when there was a male experimenter giving it to them versus when there was a female experimenter administering this task. And so when they did that, they were more likely to lose money. They were taking more risks pumping up the balloon. But keep in mind, that's not the same thing as measuring ventral medial prefrontal cortex in the scanner. They want to use the reversal learning to try to say that this is all about ventral medial prefrontal cortex being shut down by an attractive female. And even if I want to try to say this is more about a poorly developed prefrontal cortex, I can't really say that from looking at reversal learning here only. It would have to also involve something to do with, looking at a brain scan. Now, another thing finally to notice is that testosterone mediated the gender relationship to aborted tricks, but interestingly, reversal learning did not. So key thing is that they're trying to say it has something to do with the way that they see this attractive female, that a reversal learning is actually what's causing the guy to take more risks, but they didn't actually find that what they did on the reversal learning mediated that relationship between the experimenter's gender and a number of aborted tricks that they did. And finally, just one more sort of interesting methodological issue is that they didn't actually measure the participants' sexual orientation. It just assumes that all these participants were heterosexual, and so we don't know. Oh, and finally, I should just say one more thing. I still think this is sort of a problem too for the study, is that no women were tested. And so we know, for instance, that young adolescent women or young adult women are more likely to take risk when they have higher levels of estradiol, especially at different times of their menstrual cycle. So perhaps all of this would have worked the same if you had found women skateboarders doing this. And it would have been all related to something about elevated estradiol and risk-taking. But we don't know because no women were tested here, so it's just something to keep in mind as you think about the results of this study. All right, so that is a little bit of a sample of risk-taking in social neuroscience for adolescents. Now let's look at this topic, social acceptance and exclusion. And this is something that Sapolsky talks a lot about in his chapter. So I want to just review with you a little bit about exclusion and rejection. And we can kind of think a lot of, thank this guy for a lot of the research that's been done on this topic about what happens when we are rejected or ostracized. Interestingly, this is a place where Australia had its mark in the sense that Kip Williams, who's the guy in the picture there, started doing this interesting work on ostracism while he was at UNSW, and then he moved to Macquarie. But now he has moved to the United States, and he's at Purdue University, where he's an esteemed professor. So it was really Kip Williams who really began this sort of modern era of research on rejection, or social rejection and ostracism. So what I want to do next is just play for you a little video excerpt from a video that was made over 20 years ago now, but I think it gives you a good idea of like what Kip Williams was doing, how he thought about this kind of research at that time, and then I'll go ahead and tell you about some of the neuroscience research that followed.
Psychological research confirms that from birth, human beings have an intense need to belong, to connect with others in enduring close relationships. Clearly, children's attachment to their caregivers promotes their survival and well-being. When adults are asked what is necessary for their happiness, the most common answer is satisfying close relationships with family, friends, and romantic partners. People spend much time thinking about actual and hoped-for relationships. When people are ignored, excluded, or rejected, negative emotions can be overwhelming. Dr. Kipling Williams and his colleagues at the University of New South Wales have been studying the phenomenon of ostracism. It affects both those who use and receive this interpersonal strategy.
Ostracism is the act of excluding and ignoring an individual or a group by an individual or a group. And it has a lot of names. Sometimes it's called shunning, exile, banishment. Those would be cases where you are physically removed from other people. But sometimes you're in the presence of other people, in which case you might call it the silent treatment, getting the cold shoulder. In England and in Australia, they call it being sent to Coventry. The Amish community call it Maidung, where they ostracize somebody from within their group when they violated some norm.
Ostracism threatens 4 fundamental human needs. The need to belong. Human beings need to feel connected to others. Obviously, ostracism undermines this feeling of belonging. Sense of control. People need to feel in control of their lives. Even in conflict, people can exercise some control as long as they can argue their point of view. When ostracized, they lose that sense of control. Self-esteem. Human beings need to value and respect themselves. Ostracism produces a feeling that an individual has done something wrong, or that there is something about a person that is different or bad. Sense of meaningful existence. People need to feel that their existence has purpose. Ostracism takes away that sense of purpose and gives a person a look at what life would be like if they didn't exist.
Ostracism is a metaphor for death. This is what life would be like if you didn't exist. And so you feel yourself as though you're invisible and not there and totally meaningless. I was at a park one day with my dog, and I was just minding my own business, and suddenly a frisbee knocked against my back, and I looked up and I saw two guys that were playing with the frisbee, and I tossed it back to them. And to my surprise, they tossed it back to me. And I felt kind of good for a moment and thought, well, this is fun. And I got up and went and joined them, and we were tossing the frisbee amongst ourselves for about 3 or 4 tosses. And then suddenly they'd stop tossing it to me. And I'm standing there, and they're tossing it to each other, and they're having fun, and I'm feeling particularly awkward and strange and not knowing what to do, and I felt bad, and finally I sort of backtracked to my dog and licked my wounds. But I thought to myself, gee, that's a really powerful and effective way to produce this feeling of ostracism that I could possibly do in the laboratory.
In Williams' experiment, one subject and 2 Confederates are instructed to wait quietly in a small room for the experimenter to return.
People, it was like for learning experiment or like teaching how people learn or something like that. But it was so bizarre because the people weren't actually shocking anybody. Like the people, they were like behind some kind of glass thing, and they were just acting like they were being shocked, but they weren't being shocked. But the people.
One of the Confederates discovers a ball and begins tossing it.
Wow, it was bizarre. It was like, and they would keep on increasing the strength of the shock, and it was like, they increased the strength of the shock, and they thought they were hurting this person, but they weren't.
Eventually, the Confederates stopped throwing to the subject. In this reenactment, the subject clearly shows signs of stress as the ball tossing goes on without her.
And I don't know, I thought it was really weird. It was bizarre, I can't imagine. Can you imagine shocking someone? Yeah, it was weird. I thought it was funny.
So I guess we're supposed to talk about these questions. So this first one's about divorce and why we think the divorce rate's so high.
In a similar experiment, 2 Confederates and 1 subject seemingly comprise a discussion group. The reaction of the subject is easily observable as she discovers she's being left out of the conversation.
I don't know, it's kind of different. Maybe it's harder to meet people now than...
Did I see you by the Parliament the other day? Playing basketball?
Oh yeah, I'm over, I don't live there, but I live in Corbett, and I know friends at Parmalee and stuff, so sometimes I go hang out there. I have courts over there too.
I live in Parmalee.
Oh, yeah?
Food's terrible.
Really?
Yeah, it's so bad. What kind of carpet do you have? Mine's really bad. It's got like holes in it. Burns. Cigarette burns.
Yeah, the dorms, I don't know, they're not all that great. I can't wait to move off campus next year. That'll be nice. Have your own place, no one to kind of tell you what to do, and you know.
Yeah, I gotta find some people to find a place with because I can't deal with the dorm.
No, my roommate really is kind of a little bit weird. You felt that little thing that says your interests and things you like and stuff like that, and you figure they'd find someone who would kind of be normal based on what you do. But this person's just really weird. He's just, I don't even understand him.
Yeah, my roommate, when he's there, he just get the music blaring, and I put on my car and I wanted a quiet room, and I got this guy.
In a third experiment, Williams and his colleagues extended the ball-tossing paradigm to cyberspace. In this environment, the subject tosses a virtual ball to 2 other people located elsewhere on the Internet. When an individual stops receiving the ball, he or she begins to feel excluded and isolated, exactly as one would in a face-to-face situation. Cardiovascular measurements taken during this cyber ostracism experiment show that the subject experiences physiological arousal and stress response. These short-term effects are similar to those of people who've been ostracized over a long period of time and have major implications for both psychological and physical health.
There's several ways to respond in the middle stage to try to regain a sense of belonging and control and self-esteem, and some of them may be more functional than others. Sometimes you try to reestablish your sense of belonging by maybe conforming to the group a little bit more, or being more pleasant to be around so that you're not going to be ostracized. But other times people respond to ostracism with frustration because they're not getting any response, and so they try to provoke a response, and they might say something or get in somebody's face, or if they still don't get a response, they might say very nasty things or maybe even shove them. And we have one case where the wife eventually, because she wasn't getting a response from the husband, picked up a marble ashtray and threw it at her husband's head. There are many times in our interviews with people who have had exposures to long-term ostracism where they initially reacted with aggression and violence. And so one way to reestablish a sense of control is to provoke and become violent so that you do control something.
All right, so now that you have a good idea of that kind of research, and at least the way it was done behaviorally, and near the end of that video, you could see that they were looking at cardiovascular measures for how people might respond to being excluded in the cyberball paradigm, we can move on to a collaboration that Kip Williams had with Naomi Eisenberger and Matt Lieberman at UCLA. So that's Naomi Eisenberger there. Matt Lieberman, remember I told you, is the guy who started up social cognitive neuroscience. And so the three of them published this paper in the journal Science in 2003. What they did was they had fMRI scans that were acquired while 13 participants played cyberball with 2 other players. I have to chuckle a little bit when I see that it was just 13 participants. That's a pretty small sample size, but I think back in 2003 it was perfectly acceptable. So anyway, they have 13 participants, 9 of them are women, and they were all told a cover story that the researchers were really just interested in mental visualization ability. So they were really interested in having them look at the stimuli that were in these things and measure their ability to visualize and so on. But obviously this was really a study using cyberball. There were 3 scans that were done during the study. One scan involved the participant watching the 2 other players play cyberball. By the way, the other 2 players weren't actually in scanners. The whole thing was, again, contrived, just like you saw in the video, where the real participant, was led to believe that there were 2 other players somewhere else playing these games. And then they played with the other 2 players. So they got to toss the ball back and forth for the entire scan. So they were just tossing back and forth. And it was like a nice inclusive type of interaction. And then finally, in the 3rd scan, they would be excluded after they had received 7 throws. So they kind of built it up. So they've been tossing the ball around between the 3 of them. And then finally on the 7th throw, the other 2 players stopped throwing it to the participant. And so now the participants meant to feel excluded. So you have those 3 scans, and then what they're going to do is contrast 2 of those scans against each other, right? And so what they did is they looked at particular areas of the brain that they thought were involved in pain, physical pain. And the 2 that they focused on the most were the right VLPFC and the dorsal ACC. And what they found was more right VLPFC and more dorsal ACC when participants were excluded versus when they were not excluded. And so they concluded that this suggested that social pain is analogous in its neurocognitive function to physical pain, alerting us when we have sustained injury to our social connections, allowing restorative measures to be taken. So basically, just like when you hurt yourself physically, your brain responds the same way through social exclusion because it's sort of dangerous again to you to have these broken social connections. And so the same parts of the brain become more active, according to Eisenberger et al. They also had some really interesting correlations with how distressed the participants felt. So for example here, in the exclusive condition relative to the inclusion condition, they found that the more distress that the person had was more positively correlated with activation in the dorsal ACC. So if you have just been in that situation, you're rating how distressed you feel about being excluded, the more you felt excluded, the more you were showing activation in the dorsal ACC. They also found that distress was negatively correlated with VLPFC. So the people who were more likely to feel distressed by social exclusion had less activity in their VLPFC. So it's like they weren't really managing their distress with their prefrontal cortex. And finally, the DACC and the VLPFC were negatively correlated. So people who had higher dorsal ACC activity during this case were the ones that had lower VLPFC. So again, it kind of shows maybe some sort of connectivity between the two things, that maybe the reason why the dorsal ACC is up so much is because the VLPFC is down. It's not going as much up when the person's feeling this kind of distress. All right, so that's the study that was done in 2003. It got a lot of attention. A lot of people have been excited about it, tried to use other versions of it. You'll find that there are other studies about this. But I want to focus here on using this methodology with adolescents. So what about social exclusion and adolescence? Well, here's a study that was conducted by Sebastian et al. In 2011. You can see the title here. It's Developmental Influences on the Neural Bases of Responses to Social Rejection, Implications of Social Neuroscience for Education. So what they did here is they used the cyberball game in an fMRI with 16 women who were 24 to 38 years old and 19 girls, 14 to 17 years old. Now you'll notice that they only used women. They only used girls. And by the way, Sarah Jane Blakemore was a co-author on this particular paper. So why do they use girls only? Well, they say in the introduction that, first of all, girls are more sensitive to social exclusion in adolescence. And so that could be one of the reasons why it's just the research shows that they're more likely to feel socially excluded. And also they found that kind of social or relational bullying is a lot higher in girls than it is in boys. So it seemed to them quite appropriate to use this sort of paradigm to look at social exclusion in adolescence. Now, they had two kinds of blocks in their study. They had an exclusion scan and they had an inclusion scan. They didn't have one of those just watching the two people play like they did in Eisenberger et al. They also had the participants rate their social distress after they had gone through each of the scanning blocks. So like how distressed they felt after inclusion, how distressed they felt after exclusion. And they also completed what's called a resistance to peer influence measure. So this has a bunch of items, and you're supposed to indicate how much you agree or disagree with these items as it refers to you. So an item might be like, some people go along with their friends just to keep their friends happy. So if you're somebody who agrees with that, you're showing less resistance to peer influence, right? Because you're showing that you're really affected by peer influence. And it turns out that on that measure, the adults in this study were higher on the measure than adolescents. I guess that makes sense, right? So adults have high resistance to peer influence relative to adolescents who are going to be much more influenced by their peers. But there's also some individual differences here, right? So we're going to have individual variation on this measure. And not all of the adolescents, for example, are going to be low on resistance to peer influence. So what did they find in this study? Well, they looked at the same areas that had been found in the Eisenberger et al. Study. And what they found was that both groups did report feeling more distressed and negative during the exclusion condition. But they didn't find any interaction with age. So whether you were an adult or whether you were an adolescent, both groups felt distressed after they'd been excluded on the cyberball task. Just like you saw that Kip Williams was when he was talking about that Frisbee game in the park when he first got the idea to do this kind of research. He was an adult and he still felt excluded. However, across both adolescents and adults, there was increased activity in the MPFC, the subgenual ACC, and the VLPFC regions for exclusion relative to inclusion. So again, there's no age effects here, but those in people in the exclusion condition, in that condition where you're being excluded, you saw increased activity in the medial prefrontal cortex, the subgenual ACC, which is not the dorsal ACC, it's actually a little bit lower than that. And then you also have the VLPFC regions. They didn't find any effects on the dorsal ACC in this like they did in the Eisenberger et al. Study. And finally, they also found an age by condition interaction for the right VLPFC. For adults, it was higher for exclusion than inclusion, but for adolescents, it was higher for inclusion than exclusion. So what that tells us is that the adults were more likely to use their right VLPFC in the exclusion conditions than adolescents were. And perhaps that means then that during the times that they're feeling excluded, the adults are able to tell themselves, hey, it's just, cyberball. It's just these two guys throwing things. It doesn't bother me. So they're using some prefrontal activity there to help dampen down their response of exclusion. Now, finally, we can look at the medial PFC and see how it's related to their resistance to peer influence score. And they found that this was actually related for the adolescents. So you can see here, the higher their resistance to peer influence, for the adolescents, they're the one that has that dark line that's going from upper left down to lower right, that the higher their score was on that measure, the less they were able to show medial prefrontal cortex when they were excluded. And therefore, it's kind of showing us then that those peers that are able to actually do a better job of managing their exclusion are using their MPFC less as when they have a higher resistance to peer influence. You notice that for adults, it doesn't really matter. There's no effects here for adults. But the greater resistance to peer influence, the less the MPFC was activated during exclusion. Now, let's go ahead and talk a little bit about the interpretation for these results. And some key things to notice is that, first of all, it is really sort of important, I think, that they didn't find that the dorsal ACC was active during social exclusion like the way they did in the Eisenberger et al. Study. But it's important to know that there was another study that was done by the Eisenberger group, a study called by Maston et al. In 2009, and they tested 12 to 13-year-olds and didn't find any dorsal ACC then either. So it could be that it's just something about the way they're doing their methods differently from the original Eisenberg study about whether or not you have all three conditions, are you using exactly the same instructions, something, but we're just not getting any dorsal ACC like they did in the original study. This study did find subgenual ACC, however, and so did the Maston et al. Study. And this might have to do with that area of the brain coding the social exclusion as a negative emotional event. That is, that any such negative social events cause more subgenual ACC. So it might not be that it's actually unique to social exclusion. It's more about the fact that something negative has happened to you socially, and so you get this increase in subgenual ACC. And it's not really about pain necessarily, or about the pain of exclusion like you found in the original Eisenberger study. And also notice that if you look across these studies that they get differences in the left or the right VLPFC. Does it really matter? Well, the authors of this paper argue that perhaps these findings are there bilaterally and just depends on the methodology. So it could be that when you have just slight variations in the way the instructions are or the way the participants make button presses, that this could cause differences. And so they kind of like almost say like, don't get upset about this. Don't worry about whether it's the left or the right VLPFC. It could just be that the methodologies are just exposing slight differences, that it's probably really bilateral activation that happens in the VLPFC. All right, so there's our third topic that we've looked at in depth, social exclusion acceptance. And so I want to finish up this lecture and our talk here about adolescence for these last two lectures. Just in sum, I would like you to kind of reconsider a little bit about what we know about adolescence based on this work that's been done in social neuroscience. So we know now a lot about how the brain and how hormones change over the course of adolescence. And so one thing I think is really important to keep in mind then when you have to deal with an adolescent or you have an adolescent in your life is to practice patience, kindness with the adolescents and understand what they're going through. So treating them like they're just carbon copies of an adult is a mistake, right? Because their brains are not functioning and developing in the same way that an adult brain is. That you want to make use of the fact that they want to take risks, but instead what you could do is you could try to encourage their risk-taking to be more about positive risks, like trying out for the team or going into this particular contest. So the fact that adolescents like to engage in risk-taking, and especially when it involves social rewards, could be used by parents and teachers to perhaps get their adolescent to try out new things that are positive. And perhaps then they can learn consequences and assert their independence from taking risks. I think importantly, I said in the last lecture, is that sleep is really a problem for adolescents. And so having them get sleep and then teaching them about things like meditation or calmness and things that help them to regulate their emotions while their prefrontal cortex is developing could also really help with adolescence. In terms of the environment and teaching, It's interesting that about 40% of children around the world leave school in early adolescence. That is, about 40% of all kids around the world are just getting out of school and while they're in their early adolescence. And sad about that is that we should probably be thinking of this time of adolescence as just as important for their stimulation the way we do about early infants who have all those toys. So you remember, you probably, those of you who've had kids would know this, but there's this lot of this activity that we have around when babies are born, that we kind of give them all sorts of really cool toys that stimulate them and have really cool patterns, and they have these educational toys and so on. There's a lot of this emphasis on early development, but perhaps we should be thinking about this the same way for adolescents, because adolescent brains are changing rapidly. They're learning things very quickly. And so giving them all sorts of great experiences should be a priority for adolescents, rather than having them leave school. Having them get involved in really cool activities might be an important thing as part of their adolescent development. And finally, I just want you to think about this, and we're going to talk about this a little bit in the workshop, what this all means legally. In terms of you have a developing brain. Obviously, one thing you could say is that perhaps governments and society and families need to protect our adolescents from sexual predation, because they're not going to have enough to deal with some of the tricks that people might be using on them. But also think about these things. Like, we give adolescents the opportunity to drive at a fairly young age, to be independent, to drink alcohol. The ages in which they're doing this are not necessarily corresponding to when their brains are fully functioning or fully developed yet. And so therefore, they could be getting addicted more easily. They could be taking risks that are going to cause their deaths more easily. It's just interesting how the age of the legal age for these kinds of activities changes in different countries and different parts of the world, even though it's biologically probably something that happens later in adolescence, that you're going to have this prefrontal cortex that's really fully come online. And finally, the last thing to do is consider a little bit about crime, about the ages at which we should consider their actions to be adult actions. Can really somebody who's 13 or 14 that commits a violent crime be considered to have the same intellectual faculties that an adult who committed that crime would be? Because again, their brain is still fully developing. They got all these hormones that are happening from puberty. And so it just makes us maybe pause and think a little bit about what the ramifications might be for criminal treatment of criminals and the laws that we have for different crimes and ages. So in sum, I've gone ahead and then made this sort of in-depth lecture on emotion processing, risk-taking, social acceptance, and exclusion. And finally, I wanted you to think a little bit about how we might think about how this could happen, how we should take these kinds of lessons about adolescence and take them to teachers, to parents, to older siblings in society, and about how we should be thinking about adolescence. All right, that's all I have for you now. And the next lecture, we'll be talking about much earlier development.